Hi, everybody. Welcome to the 2021 seminar series of the Dialogues on Ethology and Behavioral Ecology. This is yearly organized by the Ethology unit of the Department of Biological Sciences at the University of Pisa. Today is it's very exciting for me because it is the first time we are going broadcast live. And so this is unusual. And of course, this year we had to offer these kind of seminars only remotely for obvious reasons. So this is the first of 5 seminars that will be offered. And the next ones will be one every month. And the next ones will be on Wednesday 17th, when Professor Claudio Carrere of the University degli Studi della Tuscia will speak about animal personalities, the power of the individual. And then the next one will be on Thursday, 15th of April, with Dr. Sara Marshall-Pescini from the Konrad Lorenz Institute of Ethology, and she will be talking about domestication effects on social cognition, domestic dogs and wolves in comparison. And then the last one that is planned so far is on Thursday, 20th of May, with Dr. Paolo Domenici of the National Institute Research Council at the IBF of Pisa. And he will be talking about a very interesting topic on animal locomotion and predator-prey interactions, the attack strategies of large marine predators feeding on small fish. So as for today, I leave to the podium to Professor Paolo Luschi, that will be introducing our speaker today, that is Alessandro Cini. And Alessandro will be talking about the complexities of social parasite evolution, violence, deception, and rebellion. Very good title, Alessandro, by the way.
Okay, thanks, Alessandro Massolo, Alessandro the First. I am pleased to welcome here Alessandro II, Alessandro Cini, who is currently research associate at University College of London. So he's now based in UK, but he comes from Pisa, actually, where he got his graduation several years ago. And then he continued to work with colleagues at the University of Florence, where there is this big line of research on social behavior in general. And so where Alessandro has developed his main techniques about the study of wasps, especially social wasps and other insects, especially from the social point of view. I will not use much more time, just to underline that Alessandro is certainly a brilliant researcher, and for sure will inform us about most interesting topic of his recent work. And let me, so we are all looking forward to hearing this, what he has obtained recently. Let me just conclude in saying that we are at the moment trying to find a way to have Alessandro back here in Italy and in Pisa, if possible, in some way or another. So hopefully we might have this opportunity to have him here working with us in the future, but I don't want to enter in more details for the moment. So now I leave to Alessandro and thanking him again for being here and for his seminar.
Good afternoon, and thanks Alessandro and thanks Paolo for And thanks to the Ethology unit of University of Pisa for this invitation and for the introduction. I'm really, really pleased actually to have this opportunity to give this seminar, as Paolo was saying, at the university where I graduated. And it was several years ago, about 15 years ago. So quite a lot of time. So I'm gonna share my presentation now. Give me one sec. And in the meanwhile, a couple of words on my background from the scientific point of view. I'm an ethologist, so I'm studying social behavior, communication within insect societies, and I'm doing that especially on social insects, so hornet bees, but as you will have the way to see that in the next hour or so, especially on social wasps. Then, as a researcher, but also as a citizen, I'm also very interested in trying to apply the concept I'm developing and we as ethologists develop in animal behavior to tackle some of the challenges that the current era, the Anthropocene, is putting in front of us. So, for example, the arrival of alien species, so-called biological invasions, or the loss of biodiversity. And so in the recent years, I've been involved in studying one of the most dramatic invasions, biological invasions in Europe, the arrival of the so-called Asian hornet, Vespa velutina, which is a terrible predator of bees. And on the conservation side, I'm involved in studying how we can, from knowing the behavioral species which are endangered, how we can imagine and develop strategies to protect them. So, for example, habitat management strategies to protect butterflies. And on that research line, I'm working on in the wonderful Tuscan archipelago. But today I'm going to talk to you about the core of my research, my main research line, which are social insects, the fascinating social insects, and at least to me, even more fascinating, the social parasites of social insects. So species that exploit the sociality of social insects. And I hope to transfer to you some of this excitement, this fascination. This is the first aim of this talk. And the second aim is to convince you that this system, the social parasite, are a wonderful system to unlock the secrets of sociality. So being able themselves to exploit sociality, we can eavesdrop on them to understand how social insect works. And so I'm gonna bring you with me in a little journey through into the coevolutionary arm race, an epic coevolutionary arm race between a social parasite and a social insect. And I'm gonna show you results, mainly from me, but from many other colleagues, of about the last 15, 20 years. So as you can imagine, we're gonna touch several points, but I'm not gonna go into the specific details of each experiment. But clearly, I'm more than happy to discuss with you the experimental setup and all the specific at the end of the talk. So,
and Alessandro,
yes?
excuse me, you should take away the banner on the bottom of your, yeah, thanks.
It's better like this? Should be fine. Let me know otherwise.
Yes, it's okay. It's okay.
Great. Thanks, Paolo. So we start from social insects. And when we think at social insects, our mind goes, so we have to click, yeah, our mind goes to two concepts, division of labor and cooperation. Division of labor is the fact that within an insect society, we have some individuals, few individuals that reproduce, and many others, the largest part of individuals, who are not reproducing, but they're working. So foraging, defending the nest, taking care of the brood. And you have here, top right, a wonderful example represented by these by workers of honeypot ants, which have these enlarged abdomens to store food reserves within them for the entire colony. And the other concept is cooperation. For example, taking care of the brood, but also the extreme forms of self-sacrifice represented by the bee that dies when has to defend the colonies from the attack of an intruder, a vertebrate intruder. So these two concepts, cooperation and division of labor, are defining features, are pillars of insect societies. And they are likely the reasons why social insects had a wonderful and enormous success on terrestrial ecosystems. Social insects, despite they represent just a tiny percentage of insect species all around the world, about 2%, they, on the contrary, represent about 80% of the biomass all around the world. So social insects are everywhere. They are in large numbers. They weigh a lot altogether, but that's not just the reason why I'm saying they're dominant. The point is that they're occupying a lot of crucial nodes in our ecological networks. So in the terrestrial ecological networks, they do pollination, they do predation, they are decomposers of soils and nutrient recyclers. And many plants depend entirely on ants, for example, for seed dispersal. So they're really important. We can consider them the little social things that run the world, actually. And now we all know about bees and the fascinating ants, etc. So let me spend a couple of words in defense of the least loved social insect, wasps, social wasps. Basically, nobody really appreciates, not nobody, but in general, people do not appreciate the role of social wasps in our ecosystem, but actually they're really important. They provide a lot of ecosystem services. They are good biological indicators. They can, at least in some culture, provide food. They can provide useful molecules for health reasons, but especially they are big predators. And in this picture, we draw the predation network for what we currently know, the best of our knowledge all over the world, about what insect orders and what invertebrates are predated by wasps. Here you have 3 groups of wasps. So Vespine, the yellow jackets and hornets, Stenogastrine, which is a tiny group of wasps from the tropical rainforest, and Polistine. These are the wasps we're going to talk about today. And each line connects a group of wasps with a prey, and as you can see easily from this image, they eat many different kind of prey. So they can be very important generalist predators, but also efficient predators. So to give you an idea, think that the colonies of yellow jackets wasps that live within an hectare of territory within a season can eat up to 10 kilos of prey biomass. And also, like 10 kilos might not seem a lot, but if you translate it to the number of single individuals, it's millions of individual prey. So you can imagine the importance they can have in regulating the population of other animals. And that's the reason why, during the last 20 years, there's been a growing interest in evaluating the potential of wasps as biocontrol agents. So I hope I defended well the wasps, and I hope to have convinced you that actually social insects are really, really important, and they had this enormous ecological success. And the reason we said it's cooperation, mainly division of labor and cooperation. But while cooperation and division of labor are the key to this ecological success, what we're going to see today is that they can be considered as well the Achilles heel of social insects. What I mean? I mean that many species can evolve to exploit this extreme parental care, this extreme cooperation and brood care. And obviously, if you're a species and you manage to do that, you save a lot of time and energy in caring for the brood. And I'm talking about the social parasites. So these species that evolved specifically to exploit the host social system, and in particular the parental behavior, so the fact that they care for brood. So they're exploiting other species that whose workers are programmed to take care for the brood, and they enslave them in order to save the energy of that energy-demanding task. So compared to what we are more used to see, the common endoparasite or ectoparasite, instead of using the physical resource of the individual, they're using the social system of the individual. And the clear-cut example of that probably many of you know, is represented by slave-making ants, also called dulotic ants. In this species, the workers of the parasite species make raids to host colonies to steal, to kidnap the brood, the pupa of the host colony. They bring back the pupae into their nest, and when the host brood will emerge, will eclose, it will work for the parasite. This is probably the most known example, but there is another kind of social parasite which is less known, but is far more common. And these are the inquilin social parasites. In this species, the parasite is dependent on the host colony for the entire life. So they have to establish colonies, the social parasite enter established colonies, replace the host queen, the former host queen, and reproduce at the expense of the host. Okay, so they have to integrate into the host society, and this is a big challenge, clearly. And this lifestyle is quite common, as I told you. It evolved several times in ants, bees, bumblebees. In this picture, you see the slice of each slice, the area of each slice is proportional to the number of species of that subfamily that shows this behavior, this lifestyle. And the number in brackets, it's an estimate, a conservative estimate of the number of evolutionary independent origins of this lifestyle. And it's about 50 times that it evolved independently. So it really pays, probably, from an evolutionary point of view. And it's also exciting because you can make a lot of comparison, across different clades. Now, as you can imagine, this lifestyle means that there is a tight, strong relationship between the parasite and the host. Okay, so the parasite needs to conquer the host colony to get the fitness, and the host colony, when it is parasitized by the parasite, is experiencing a great reduction in its fitness. So this is putting pressure over evolutionary time on both parties, leading to a coevolutionary arm race. In this coevolutionary arm race, both parties evolve adaptations and counter-adaptations in order to fight to each other. And this leads to what is considered the red queen hypothesis, a red queen situation in which you have to run faster even to stay in the same place. Otherwise, all the world around you will adapt against you in the framework of social parasitism. And indeed, social parasite shows a lot of astonishing modifications, morphological modifications, physiological modifications, in order to evolve strategies to better defeat the host. For example, enlarged glands for the secretion of certain chemicals, or increased physical weaponry, so enlarged head, enlarged mandibles, longer sting, etc. So today, I'm gonna bring you with me in a journey in one of these epic coevolutionary arm race. And as you might guess, there are two characters in our tale, the host and the parasite. So we're gonna know each of them a little bit better before starting with the more experimental part. So first of all, these two, as you can see, they are both wasps, social wasps, paper wasps of the genus Polistes. And Polistes is a genus that is well known in the field of animal behavior. It's a key genus to understand animal behavior. There are about 200 species all over the world, more or less. But the main point of this genus is that it's really easy to work with these wasps. So compared to more complex societies of, for example, bees or ants with millions of individuals inside the nest which are underground, so which are really complex to be studied. These wasps make these small colonies, so 50, 100 individuals, that does not have an envelope. The colony does not have an envelope, so you can immediately see what's happening on the nest. The individuals are quite big, you can mark them, so you can actually follow all the social interactions, which is crucial for animal behavior studies. And basically you can reconstruct a sort of Games of Thrones or Beautiful as a fiction of the interaction among them. And additionally, you can, they're quite big, so you can do quite easily some physiological measurement, for example, reproductive physiology or brain anatomy. So they're a good model. they're interesting, they're a good model, and that's the reason why probably they have so many firsts, so some records in the field of animal behavior, and not only. And I just want to give you one of these, talk about one of these, which is that it's in this genus and in one of the species of this genus, which is Polistes dominula, the host species we're going to talk about, that the dominance hierarchy, the existence of dominance hierarchy in an invertebrate has been discovered for the first time. About 8 years ago, Pardee discovered that within a colony, females were fighting to establish a dominance hierarchy, which at that time was not known for any invertebrates. And this finding was so important, at least for our community in animal behavior, that is now standing, this representation of that is now standing on the logo of the Italian Society for Ethology, and possibly, likely also inspired what is considered to be the most, or one of the most ancient jewel pendant inspired by animal behavior. And it's called the Malia pendant. It's been found in Crete, date back 1,000 years ago. And apparently, it has been interpreted as representing two wasps which are dominating each other. These lines here should be, according to some authors, the antennae which are touching one to another in order to dominate. So it's a very important genus. So we start from the host species. We need to know how the host species is behaving and its lifestyle in order to understand how the social parasite can exploit this system. These wasps are annual wasps. So the colony, contrary to what we are using in other insects, which are perennial, like honeybee colonies or many ants, in this species, in Polistes genus, the colonies are founded in spring and they end at the end of the season. They end at the end of the summer. So they're founded around April by one or more females. In the case of more than one females, these females start to fight to establish the dominance hierarchy, and only the top dominant individual will become the queen. And this is important. So there is a high reproductive skew. One individual monopolizes reproduction, the others work. So if you want to get the share, if you want to reproduce on a nest, you need to become the queen, you need to become the top dominant individual. So after the colony has been founded, all the eggs are laid and workers emerge. The workers take care of the colony, they defend the colony, they forage, they feed the brood, and then they also feed the reproductives. So reproductives are males and females, which will not work on the colony, the females, but they all leave the colony. They will mate, and then the males die, as usually happens in social insects. They are so they've been considered like a flying packet of sperms, because basically their unique role, apparently, is just to fecundate the female. And the female will spend the wintry season in sheltered places. So those mated females who will survive the winter will found new colonies the year after. So this is the life cycle. And 2 points are important to be stressed here. One is the dominance hierarchy. So as I told you, there is a dominance hierarchy which rules reproductive skew on the nest. And the second point, which is again different from what we are used to when we think at social insects, is that here workers are fertile. So in bees colony or in many ants colonies, workers are sterile. Here, the workers are fertile, which means they can develop ovaries, they can mate with males, and they can lay eggs on the colony. So in principle, they can have their direct fitness. And in many cases, they can also replace the queen. And this obviously creates this substrate, the ground for many conflicts over who is going to reproduce, okay, and who's going to work. So it's a more plastic society than what happens in many most famous insect societies. We now go to the parasite, Polistes sulcifer. So this one is the social parasite, and it's one of the cases of obligate social parasitism, which means that the parasite has lost the ability to found its own colony. If you take a parasite and put in the lab with all the materials to create a colony, it will not create a colony. It will just wait to usurp a colony. So no way to do that unless it usurps a colony of the host. And also, it has lost the ability to produce workers. It's just producing reproductives. So again, even if it colonizes a colony, but without the host worker force, no way to get the fitness, okay? And it's one of the 3 cases of obligate social parasitism in Polistes genus. They're all related here. As you can see, they're closely related to their hosts. And each of these parasites has a different specialization or generalism in terms of the host they have to usurp. So our case, Sulcifer versus dominula, is the case in which Polistes sulcifer is the obligate parasite of its only host, Polistes dominula. So here again, you see the strength of the selective pressure that must be on both these parties. Now, the life of the social parasites, the life cycle, starts in a way a little bit later than that of the host. So the first difference is that the social parasite is overwintering, the females are mating and then overwintering at the top of the mountain. Here you can see a nice picture of Monte Sibillini. Here is about 2,000 meters. And so here should be like 1,700, something like that. And here is where we're going to collect the social parasite when at the end of the overwintering phase. So they overwinter under these rocks. And obviously, at the top of the mountains, the spring comes later, no? And so the overwintering stage ends later. And so when the parasite, the females of the parasite, exit the overwintering stage, it's already advanced spring, and they then perform altitudinal migration toward the lowlands where they look for the host nest. And the host colonies at that time will be already started, okay? So they arrive around the host colonies when they're already quite well developed, before the emergence of the workers, but with colonies already started. And here you have a picture of some of the lowlands when we can look for these parasites who are trying to usurp. This is the wonderful Piana di Castelluccio di Norcia. Actually, the picture had been taken during the blooming, so it's July, so it's a little bit later. It was a very nice picture. And so when they arrive in the lowlands, they look for the host nest. In our places, the host nests are usually made under these tiles on the roof of the houses, which are very warm and sheltered places. And the parasite arrives there, look for a host nest, recognize the right host nest of the right colony, the right species, and then conquer the colony. He conquers the colony with fights. It replaces. It can either kill or just dominate the former host queen, replace it functionally, take the leading role in the colony, and enslave the workers. Then the parasite the brood of the parasite has a shorter developing time compared to the host, so the cycle of the parasite is a bit smaller, shorter. Reproductives will emerge, they will leave the colony, mate, and go to the top of the mountains. Okay, so these are the two life cycles, well overlapped. And today we're gonna see this little tale in three. We're gonna divide this little tale in 3 chapters, which will be of unequal length depending on the extent of our knowledge about each of these phases. The first chapter will be about the conquer of the host nest by the social parasite. So is the social parasite able, and to what extent and how he manages to conquer the host nest? The second chapter is about how to exploit the host society. So the first line is to usurp. The second part is to integrate, infiltrate, get accepted into the host colony. And the third chapter will be about the possibility that the host species is not so happy about that, and possibly how they react. So we start from the first part. So for any of these parasites, the first important part is to infect a colony, to usurp a colony, to overcome the first line of defense. And just to give you an idea, you have to imagine that a colony of social insect is a very, very well-defended and xenophobic fortress, okay? They do not like intruders at all. They repel fiercely an intruder who tries to go into colony because they want to steal something, or even because they just get lost, okay? So they're really, really ready to aggressively react to intruders. And here you have a short video. It comes from one of our experiments. We were evoking the aggressive reaction of a resident foundress that you can see here, this one, with the presenting an intruder. And in this case, the intruder was a lure of a dead wasp. So it was not moving, but it was keeping all the chemical and visual cues that elicit in the host species a reaction. This is a lure of the same species. We are not talking about social parasite. It's a general intruder. And you can see the bites. The wasp tries to sting, tries to cut legs. So it's a very, very harsh reaction. So imagine that the parasite, the social parasite, has to overcome this line of defense, and usually not only of just one wasp, but 2, 3, 4, 5 wasps together. How can this happen? There are two possible strategies that have been well described in the literature for other social parasites, which are basically deception or fight. So a social parasite might try to not get detected, not being detected, or at least being detected but not as much, not a lot, or simply saying, okay, I'm going to be detected, I'm going to fight. So violence on one side against deception. And we have indications for both of these strategies, that both of these strategies can be important for our social parasite. So we wanted to understand which one is playing the leading role. And regarding the first strategy, when an intruder arrives to the colony, it is usually spotted thanks to its smell. So the resident wasps are feeling the odor, are perceiving the odor on the cuticle of the social insect, of the intruder. And this odor is made of a blend of cuticular hydrocarbons, many compounds, with a complex mixture of compounds. Each colony has its specific odor, and so they can compare their inner template of the odor with the odor found on the intruder. So one possible strategy is to arrive at the nest without these cues, okay? Or at least with a very low amount of these cues. So adopting what has been called chemical insignificance strategy. And indeed, this strategy is present in the two other species of social parasites of this genus. So, for example, Polistes semenovi and Polistes atrimandibularis, they both arrive at usurpation with a very low amount of these compounds. And thanks to this, they go unnoticed in a way, at least less noticed than it would happen having the entire odor, okay? So our idea was, okay, that from the same genus, it might happen, and also our parasite is using the same strategy. But at the same time, we also know that the parasites, when they arrive to the host colony, they fight a lot. And also, during evolution, they evolved enlarged heads and bigger bodies and strengthened mandibles. So basically, it seems that they have a morphological adaptations to fight. So we wanted to weight the two strategies. And to do that, we decided to quantify the amount of cuticular hydrocarbons, all these molecules on the body, and to compare these within the social parasite, trying to understand if it has less of these compounds compared to the host. Now, we know that the social parasite during overwintering has the same amount of compounds, of these compounds, compared to the host. They have the same level, probably because they are needed, these compounds, not only for communication, but also for avoiding desiccation, for preventing the arrival of parasites and microbes. So they have the same level. So we were expecting to find a decrease in the level of cuticular hydrocarbons in the social parasite at the moment of usurpation, when it's needed to smell less, okay? So to do that, we use gas chromatography and mass spectrometry analysis to quantify the amount of these compounds before usurpation and comparing before usurpation with the usurpation period. And the answer is that this social parasite, contrary to what is known for the other two species, is not using this strategy. So they're arriving at the host nest with the full amount of smell. So they can be smelled, they can be perceived and recognized, okay? And in this graph, you see that this chemical significance is not present for any kind of different chemical compound class we can analyze, okay? So we're quite sure about that. And so we have to go to the other hypothesis, fighting. And if you record the interaction at usurpation, this is what you can see. These are, one is the host and one is the parasite, and they fighting, they're trying to sting themselves. And here you can see a leg, a piece of a leg, which belongs to the parasite. You can see here, this is the piece lacking from here. So they're really harsh, these fights. Nonetheless, despite there's one leg less, the parasite arrives on the nest and is trying to conquer it. So fighting is really important. So we put our attention to fighting, and we were interested in understanding which is the factor that predict the success of the social parasite. And so we staged in the lab usurpation trials, inserting one social parasite into a cage where there was a colony of the host, okay, and we recorded the fights, and we recorded the success of these fights. And it came out that actually it's really size, simply, it's the size that matters during usurpation. So rather than deceiving the host having not the smell, it's really being bigger that is important. So size matters a lot here, and successful parasite, those that won the usurpation, were bigger than unsuccessful ones. So we can say that for this first step, the usurpation step, fighting is important rather than fooling. Now, the story is never so simple, so actually there is also a little bit of fooling, because actually the parasite is able to release an odor which elicits aggression in the host, but intraspecific aggression. So it's fooling the host species by making the foundresses to fight against each other, okay? So there is this sort of propaganda substance, as it has been called. But apart from that, there is also a lot of fighting, as you saw. And when the parasite then arrives on the nest, he still has to dominate the other foundresses to make them convinced that he's now the dominant individual. So the first step is done. It seems that the parasite is quite good in its job in overcoming the first line of defense. But overcoming the first line of defense is one stuff. A completely different stuff, a completely and more difficult challenge is to integrate, to infiltrate into a complex system, such as a colony of social insects. You have to imagine that within a colony, there is a continuous exchange of interaction, dominance interaction, food exchange of information. So social insects within a colony, the nestmates, are continuously cooperating, exchanging chemical communication. We're gonna see also vibrational communication. So it's really a complex system. How can the parasite arrive to integrate properly into this system and exploit the host workers? So we decided to see, we're gonna see today these three possible lines, three possible landscapes, let me say, in which the social parasite has to integrate in order to exploit the host colony. And the first, more evident one is the chemical communication system. So I told you before that there are these cuticular compounds that are important for the social insects, and thanks to them, they can recognize the species, the colony, colonship, and also different roles of different individuals within the colony. So they're really important. And several years ago, when I was still at high school, I guess, there has been a lot of work on the chemical integration of the social parasite within the host colonies, made by the group that Paolo was citing before, the Wasp group in Florence, led by Stefano Torillazzi. And they discovered a very interesting stuff, a very surprising stuff, that the social parasite is able, in a very short time frame, to mimic the odor of the host. So here you can see a chemical profile of a pre-usurpation parasite, so before usurpation. Here you have the different compounds with their abundance, and this chemical profile, this signature, is different from that of the host. You can see many different peaks here, different abundances, so they're different, they smell different. But just 3 days after usurpation, the parasite is smelling as the host colonies. And the parasite is not only able to acquire, we still don't know if it is synthesizing or possibly acquiring the odor. Probably the second option is more true, but we still don't have data. But anyway, whatever the mechanism, within 3 days, the parasite manages to get the same odor of the colony and the same odor of the former host queen. Okay, so it smells like a queen. It's perfectly integrated. So, okay, good job for the chemical communication part. But what about the two other lines? Okay, what about behavior? I told you before, within a colony, there is a integrate social network of interactions. So we wanted to see if when the parasite arrives, it is able to take the same place, position in the network as the former host queen had, okay? And to do that, we use social network analysis, which is a technique which is now basically very crucial, fundamental for any study in social behavior. And as I told you before, it's quite easy to do this kind of studies in wasps. So we took several colonies of control colonies and parasitized colonies, and we recorded them after having marked each wasp individually. So we could recognize each different individual, and we could record all the interactions, the pairwise interactions, in order to reconstruct a social network. And here you have an example of a social network. On the upper line, you have a social network of a host colony, so a non-parasitized colony, and below the representation of a parasitized colony, okay? So each circle is a wasp, and each arrow connects a wasp with a wasp who is doing interaction with a host who is receiving that interaction, okay? The size of the circles is representing the activity of the wasp, so how many nodes this wasp is interacting with, and the size of the arrow is representing the intensity of that specific pairwise relationship. So for example, here you see that wasp 1 is interacting a lot with wasp 2, far less with wasp 7, okay? And the color are different according to the phenotypes. So here you have workers in blue, these are subordinate foundresses, and this is the queen. And by looking at this network, you can immediately realize that the queen is central, has a preponderant role in the network, because it's the individual who is most connected, is most central, if we speak in the terms of social network analysis. So our point was to first understand the different position of the different phenotypes, queens, workers, subordinate foundresses, in the host colony, and then see, again, this is the leitmotif of this talk, is the parasite able to integrate, to acquire the same position of the host queen? And when we, these are preliminary results, actually, we are working on them, but already from our preliminary results on centrality, we can easily see that as the host queen is far more central in its social network than the subordinate individuals, here below, also does the parasite queen, okay? So the parasite queen seems to replace well the former host queen within the social network of the host colony. Okay, so well done, parasite, good job again. So 2 out of 3 challenges are well done, and we move to the third one. The third one is about a communication mode, a sensory channel to communicate, which has been really neglected in social insects, actually. It really received not a lot of attention, unless the very last years. And this is probably due to the fact that over the last years, our technological equipments improved a lot and far more cheaper. So many more and more researchers are starting to realize that it's possible to study this vibrational communication. And what is vibrational communication? An insect can produce vibrations by moving parts of its body and transmitting them to the substrate. And then these waves of movement are transmitted along the substrate, okay? And this happens a lot in social wasps. We did this review, and we, thanks to Rachele Nieri, who did a wonderful job here in describing all the different kind of movements that can produce vibrations on the nest, okay? So the wasps can tremulate, can drum, so beat, for example, the antennae on the nest. They can scrape the nest surface. So there are many different kind of behaviors that produce vibrations on the nest. And why? For many different reasons. For example, you can see here for defense or for recruitment of other individuals, for dominance, which is highly interesting to us, and for mediating a sort of discussion, a talk between adults and larvae. And these kind of substrate-borne vibrations are well spread in Polistene. So here the question is clear. What about our host species? Because if there is a vibrational landscape in the host species, the parasite needs to integrate in that vibrational landscape as well. So we wanted to understand this, and we focused on this behavior that I'm gonna show you, but you have to hear, so I'm gonna be silent for a while. This is called abdominal wagging. It's a shaking. You will see lateral shaking of the abdomen, which touches the surface from time to time of the nest and produces vibration. And what you're gonna hear now is the amplification of the physical vibration of the nest. So we have an instrument, we borrowed an instrument, which is called laser vibrometer, which can record these movements and transmit it to our speakers, and so we can understand. Then we can also analyze them properly with the specific softwares, but it's nice to hear. And obviously it was really fascinating when we heard for the first time the vibrational landscape of a social insect. So please hear this sort of okay, so it was this that we heard in the second part of the video. Okay, so why they're doing that? There have been several hypotheses. We wanted to understand if this was important in the social life of the host species, and so we recorded these vibrations, and then we developed an instrument which is called a shaker to be able to play back these sounds. I call it sound is a vibration, but just to understand between us. So we played back these vibrations. So you have an MP3 controller, which sends the signal here, and the signal is transmitted to the nest. And here you have the individuals of the host species. And it has been said several times, but nobody really ever tested that this abdominal wagging is a signal that adults send to larvae in order to prepare them for receiving the food. This abdominal wagging is especially done when the wasp come back from foraging and is inspecting all the larvae. And so people were saying, okay, it's probably a signal to talk to larvae. But there were no experimental evidences, just correlative evidences. So thanks to this shaker, we were able, and it was really, really a great pleasure, actually, it was really exciting, to show that actually the larvae are responding to this signal. So when we played back the signal, and only to specific vibration, the abdominal wagging specific vibrations, the larvae were increasing their movement. They were moving something like that, so laterally and up and down. And especially what is important also, not only that this means that they perceive the signal, but also the more they move, the more likely they were to be fed. So this link the signal of the host with the response and a benefit for the larva. So This is to say that this vibrational landscape is very crucial for the host species, okay? And another point is that several authors, even if here we have not yet been able to demonstrate that, but there is a lot of correlative evidence that it is linked, this abdominal wagging, also to the dominance hierarchy signaling. So it might have something to do with being dominant. And indeed, dominant individuals perform this abdominal wagging more than subordinate individuals. So it's really crucial for the social parasite to integrate into the vibrational landscape as well, like the chemical or behavioral landscape. So this is a general leitmotif, and in order to understand that, we decided to compare the vibration produced by the social parasite with that produced by the host. But first, obviously, we need to know if the social parasite is producing any vibration, and actually it does. Here you can see a sort of abdominal wagging, which has been called, however, abdominal drumming, and you will understand why. Again, you have to hear that. Okay, so I guess it's quite evident that I'm producing vibrations. Again, what you hear is an amplification of the vibrations from the nest, even if in the case of the social parasite, sometimes if the lab is really quiet, you can perceive, you can hear. So there's also a component that you can hear as a sound. So we wanted to understand if these vibrations produced by the social parasite allow the social parasite to integrate into the host colony. And to do that, we need first to understand what's happening in the host colony and then compare the two species. So we recorded and compare several features of these vibrations, and we focused on several colonies of parasite and host, and we focused on some parameters. Now, here again, there are still unpublished results, and we are finishing this data. So today I'm going to present to you just one parameter for each class. For example, for the temporal patterns, we're going to see the duration, compare the duration of the vibrations. We're going to compare the dominant frequency, for the spectral features, and we're going to have a look at the intensity of these vibrations. And the first part was to compare, to understand what's happening in the host species, especially because I told you before that some people suggested that this abdominal wagging in the host is related to dominance, no? So we wanted to compare the signal in dominant individuals and subordinate ones. And what we found is that there is no differences in the dominant frequency. So from the spectral point of view, the signal is the same in dominant and subordinate. There is no difference in duration, but there is a clear difference, a significant difference, even if not an astonishing difference, in the dominant, in the intensity. So the vibrations of dominant individual are more intense than those of subordinate. So we come to the important part now. What about the parasite? Regarding the frequency, there is no difference again. So the vibration of the parasite looks like, from the spectral point of view, as the vibration of the subordinate, of the host species, subordinate or dominant. But, and here comes the really interesting result, regarding duration and intensity, we have a completely different picture. Both for intensity and duration, the parasite is significantly different in the vibration produced. So the signal produced by the parasite is significantly more intense and longer than that of the host species. And especially if you see, it's clearly even more intense and longer than that of the dominant host, so of the queen, the former queen. So here you can see there's a sort of mix of sort of mimicry, no? For the frequency part, the signal, the vibrations are very similar, but also probably an amplification. So for intensity and duration, the parasite is probably amplifying, I would say, at least physically, at least, not with any necessarily any biological sense, yet, because we never tested that yet, unfortunately. But at least physical is amplifying the intensity and the duration of this vibration. So, okay, good. The parasite seems to be doing a very good job for all these three, for these big challenge divided into three challenges. And so this could be a sort of the happy ending of our story, no? Violence, cheating, deception, happy ending for the social parasite, which is probably ahead in this coevolutionary arm race. But there is always a but in life and in science, and the story does not end here. So far, we mainly focused on the parasite side or the host queen side, but we risk to forget about somebody else, the workers, the host workers. If you remember, I told you that contrary to many other social parasites, host workers can develop ovaries, they can mate and lay eggs, so they're reproductively potentially active. So they might disagree on just working for the social parasite, and they can, in principle, react, trying to pursue their own direct fitness strategy. This was not actually really considered, and so when I was doing the PhD, I was in the position of having a lot of different nest colonies of the host and the parasite for other experiments, and so it was by hazard that I decided to cut them, dissect them, and see, just control that their ovaries were not developed, as we were all thinking, all predicting. But when I did that, it came out a completely different result. Actually, the workers of the host species in parasitized colonies, they are developing ovaries. So they have more developed ovaries than workers in the normal colonies. And if you give them the possibility to lay eggs, they will be ready to lay eggs. So they are priming themselves for reproduction. They're ready to reproduce. But this reaction, this rebellion, that we can call it rebellion, is a covert rebellion against the social parasite. Because if you look at the colony, you will not spot anything, really. If you just look at the foraging, etc., you're not spotting any difference. But if you dissect them, and these take place 6 weeks after the usurpation, they develop ovaries, they're ready to lay eggs. So there is a rebellion. Now, this rebellion can be interpreted in many different ways from the ultimate point of view, the evolutionary point of view. It might be that the parasite is letting them prepare for reproduction, as the life cycle of the parasite, as I told you in the beginning, is a little shorter than that of the host. So, or it might be that the parasite does not have a complete control. For example, the parasite might not be perfectly integrated. So it's important to understand when this covert rebellion really take place, okay? And we knew at the time that 6 weeks after the usurpation, the workers were reacting. And we wanted to know what's happening before. So 6 weeks is, 1 month and a half is quite a lot of time. What's happening just right after the usurpation? Are they already starting to react or not? We did the same experiment 2 weeks after, and there was no difference. So we were thinking there is no reaction early after usurpation. But one thing is physiology. Another thing is behavior, or, for example, gene expression. So there are things that happen first before any evident, apparent physiological change. And so we decided to go in search of these potential early markers of reaction. We decided to couple social network analysis with transcriptomic in order to see if something was happening in the brain and in the fine structure of behavior of these workers. So we did again the same kind of experiments, comparing what's happening in usurped colonies and controlled colonies, but this time we were checking what was happening in the first 2 weeks after usurpation, okay? And we were looking at dominance network analysis, so it's the same analysis I explained to you before, but just focused on workers. And we quantified brain gene expression. Obviously, the behavior of an individual is the result of interaction genes per environment, and among genes, the genes of all the body. But obviously, those in the brain are probably the first candidate you're gonna look at to understand possible influences on behavior. So regarding the dominance network, we did as before, as I told you already, and our prediction was that the workers parasitized by the social parasite would be more central than the workers of the host species when it is not parasitized. And if you remember, the dominant individual have higher centrality, and then the workers are more marginal. So we were thinking, if they're reacting, they're probably dominating each other, assessing each other to be ready to take the place of the dominant individual or start to lay eggs. So there will be less marginal. So they will have an increased centrality. For what concern the brain gene expression analysis, we quantified the amount of expression of 5 genes, which were, we considered them to be candidate genes indicative of a reproductive phenotype. So some genes were more expressed in that genus for the few information we had at that time. They were more expressed in queens, and in other cases, for other genes, these genes were more expressed in workers. So we could polarize, worker-like expression in genes and queen-like expression in genes. And at that time, we only analyzed 5 candidate genes because of some limitations in the technique, findings, and the knowledge of candidate genes. And our idea, our prediction, was that parasitized workers were down-regulating the worker-biased genes. So those genes that are more specific of workers, since these workers are reacting against the parasite, they will be less expressed. And what we found, so here you have the results concerning the dominance network analysis for outgoing centrality and incoming centrality. So this means how much an individual is central considering what it does, so the outgoing interaction. And here you have the opposite, so the incoming interactions. And as you can see, while during the first week after usurpation, there is no difference between parasites, worker from parasitized nests and workers from control nest, after 2 weeks, there is a clear difference, a significant difference, which tells us that workers in parasitized colonies are interacting more, are dominating each other more, doing more and receiving more than workers in control nests. Okay, so something is going on there. There is no more a really stable dominance network in these nests. And concerning the gene expression analysis, we found one gene which is downregulated in parasitized workers. So this gene is called imaginal disc growth factor 4, and it's molecular, it's involved in the cuticle formation. But what we know from the previous results, previous studies, is that it is upregulated in workers. And so the fact that it's downregulated in parasitized workers is a good indication that they are sort of leaving the worker phenotype, okay? And also, this gene is upregulated in stable and socially well-integrated situation. And again, the fact that it's decreasing expression in parasitized workers, probably it's an indication that their social environment is no more stable as before, which is a good link with what we found here in the dominance network analysis. So, what does it tell us? To conclude this tale, it tells us that the parasite is good, and we now know how, especially through violence, in usurping a colony, in overcoming this defense, then is good in getting integrated both at the chemical level, at the behavioral level, and also potentially at the vibrational level. But notwithstanding that, the workers do not agree with that, and they show this covert rebellion right early after usurpation, which leave us with a question: why? What do they recognize? What is the parasite not mimicking well in this system? And I tell you that I don't have an answer yet, and nor I have an answer to the question who is really ahead in this epic evolutionary arm race. we still far from completely answer this question. So to conclude, I want to say that in this evolutionary arm race, the social parasite has many tools, many keys to open the kingdom of the host colony, which are surely necessary, but probably, it seems, from the fact that the workers are rebelling, they're not sufficient. And also, I hope to convince you that these are a really wonderful system to understand how sociality works in general, but especially in social insects. So, social parasite, having tried to usurp and integrate into the host colony for a million years, they represent for us the keys to the kingdom. And the next things we're gonna do in the next few months is to try to go deeper in this transcriptomic approach, trying to understand with a more wide sequencing the molecular basis of this wonderful, fascinating parasitic lifestyle. And I hope that the next time you will see or you will think at a colony of social insects, you will also think that there's probably somebody around who has the keys to this fascinating kingdom. Thank you a lot, and thanks again for this opportunity. It's been really, really a pleasure to discuss about this, and I hope you enjoyed that. And obviously, we have plenty of time for questions, so I'm happy to answer to your curiosity. Thank you again.
Thanks to you, Alessandro. Very, very nice and interesting seminar presentation, and the topic is incredibly interesting, and it touches a lot of different theoretical questions, as well as very, very advanced methodologies of inquiry. So congratulations. So what I would do is I would open up for questions from the people on the YouTube channel, as well as from other people like Paolo. If you have some, just so you know, in the comments on the YouTube, you have some congratulations and on your clarity. Yeah, someone is helping us to show you directly the comments of the people.
Oh, okay.
Yeah. And so I don't see any specific question at the moment from the public. So I know that Paolo had a question. So Paolo, please go on with your question. Paolo, you are muted.
Okay, sorry. I wanted to thank Alessandro for this fascinating presentation, of course, and fascinating topic we spoke about. Well, I would have actually a number of questions, so I will not enter into all of them. One main one is probably very simple, probably may be something that I missed from your talk. You said that, okay, in the case you showed of your wasps, that your workers that start to breed, to reproduce, to lay the eggs and so on. So the very simple question, again, it might be too simple, where do they get the sperm? Do they need to mate with someone, with some males, I guess? Or do they go ahead by themselves with the parthenogenetic strategy?
Okay, so yeah, it's a good point. They can do both, actually. They can lay unfecundated eggs, which we give rise to males, but they can also mate and lay fecundated eggs. So usually, as I told you, most of the males are produced at the end of the season. So You're right when you ask that. Where are the males early in the season when the workers are ready to lay eggs? Usually there are not a lot of males around, but some colonies can produce early males. So there are always some males around. And actually there's been a nice experiment in the wild, in which experimenters removed the queen, and in about 2 weeks, the workers were already mated and ready to lay their own fecundated eggs. So they have both strategies. They can start laying eggs, which gives males, which is already a good investment in fitness, because then the mates will transmit the genes by mating, or they can look around and try to find males, and a tiny percentage of males is ready for mating also in June. So yeah, so both.
So these, let's say, precocious males, are they produced by any colony or by special colonies? I mean, because they might be very relevant, very fruitful for them to be ready to mate from the point of view of the male early in the season.
Usually, what we know is that there is a big skew in the production of early males. So there are some colonies that just produce males or produce a lot of males, while most of the colonies just produce workers and then the reproductives. Right. So the reasons behind are not entirely clear yet.
Sure.
But one thing that we should remember is that if a worker in a normal colony, but even in a colony parasitized by the parasite, mate and lay an egg, the egg will be soon eaten or destroyed by the dominant queen. So there is this egg policing in which the queen is able to recognize the smell of the eggs of other individuals and can eat it. So this is a sort of policing that controls, under normal circumstances, the cheating by the workers, if you want.
Right.
There is a sorry, Paolo. There is an interesting comment by Stefano Turillazzi. Maybe, yeah. Have a look, Alessandro. What is
enlarging the breeding cages? We have various cases in which the workers abandon a deserted nest and move to another side of the cage to found a nest on their own. Yes, Stefano, you're completely right. This absconding behavior in which the workers abandon the nest is a possibility for the reason that we are discussing with Paolo, no? Because if they can mate, they can leave the nest and start a colony their own. Now, in my personal experience, but we had small cages, and you're right here. We just saw that, I saw that once over the 3 years of PhD, over hundreds and hundreds of cages. But you're right, that might be because we were not in the field. So we should do tests in the field. But one thing to consider is that also it's also a risky strategy, because trying to get in the fitness of a nest which is already going on, is well defended, is well developed, it might be better in many cases than try to go alone to build a nest already late in the season, so 2 months after all the other nests have been founded. No, so sure. It's a cost-benefit balance, but it's a very nice point that would be wonderful to study in the field.
There is another question from someone in the audience. Maybe you have somehow answered these already. Can you double check? It's about probably.
I'm seeing that. How does workers, how they're able to produce eggs if they don't copulate with males? Yeah. So it's the point of before. They can lay eggs. All these species, all the wasps, they can lay eggs, even unfecundated eggs. And it's not a waste of time, it's not a waste of energy, because they produce fertile males, they can mate. So it's one of the ways to produce, to transmit the genes, so it's good. Or they can find a few males around. Okay. But the difference with the queens is that they have to mate, while queens are already mated. So this adds another layer of complexity, when they have to decide, am I gonna leave the nest, or am I working here and helping the others? It's a lot of challenges to be taken into account.
I have a couple of questions myself. I didn't see any other question from the public for the moment, but of course, please type it in case you have it, and we will transmit it as soon as we receive them. But I was wondering, you mentioned some kind of a odor manipulation for increasing aggressiveness between co-foundresses, right? Is there any manipulation of peacekeeping kind of behaviors that have been implemented by
you mean like appeasement pheromone, something that appease
something that is commonly used to diminish the level of aggressiveness? Is there
it's present in many other social parasites.
Yeah, okay.
Apparently here, well, yeah, yes and no. Not from the chemical point of view. The chemistry is all to mimic or to elicit this intra-colony aggressiveness, as demonstrated by Cini and Chervon with this sort of propaganda pheromone. There was an hypothesis from the same group about the importance of the facial pattern. If you saw the picture, the parasite has a different face, a different clypeus compared to the host, has a particular black band on the clypeus, and in some cases, this black band is attached to the mandibles, probably amplifying the mandibles. And this might reduce the aggressiveness. Actually, it has been experimentally demonstrated that it reduces the aggressiveness. So thanks to this visual feature, some of the parasite can reduce aggressiveness. However, we still do not know why, actually, why the host perceived that. We tried by increasing this band, decreasing, changing the shape, and we never actually came to the final answer. But yes, the face of the social parasite decrease as an effect decrease the aggressiveness. We are not sure yet if it evolved specifically for that as a sort of appeasement signal.
Yeah. Okay, there is another question by Stephanie. Yeah. How can the
parasite gain the odor of the colony? I would love to know that. Really good question. I have no answer. No, I mean, there are several hypotheses which have been made in other systems. The two possible ways of doing that is one is to synthesize directly these compounds that are important to being mimicked. So you perceive the compounds in the host species and you are equipped to biochemically produce them. The other way is to acquire this odor from the environment. So in my opinion is that the second strategy is more likely to occur, and this because it's not simply that they need to synthesize many different compounds according to the different colonies, so that would be a really difficult task. While if you just arrive in the colony and take the odor directly from the colony, it's obviously easier. How they do that specifically, again, several possibilities. One is by eating the brood. It's known that the cuticular hydrocarbons you have on your cuticle, if you're an insect, depends on what you eat. No? You are what you eat. You smell what you eat. And so by eating the brood of the host species, which happens, they can take some odors. Also, they live and they interact with medial individuals, so they can share the odor physically. And also, one very interesting stuff I would love to study is the leaking behavior. In the very first days after usurpation, the parasite is leaking a lot the cuticle of other individuals, and especially that of the dominant queen. And so possibly this is a way through which they
Did you see them leaking themselves after?
They do grooming after. They do grooming. So there might be a way to spread the odor. I guess there is not a single hunter here. There is a mix of possible ways to take the odors from the aliment, the feeding the larvae, and possibility.
Okay, we have another question. This is a methodological question by Carolina Signorelli. What kind of tools do you use to measure these vibration signals?
Yeah, so there are many different tools. What we use is a very expensive tool, which is called laser vibrometer. So it's a laser. They explain me that the people expert in this, they explain me that it's similar to what is used to when the police want to make you a fine because you're going to have a high speed, no? So basically there is a laser which works on a Doppler effect, and it's pointing toward a piece of paper, silver paper, that we attach to the nest. So imagine that this is a sponge, it's not a nest, but anyway, imagine this is a nest, and you have a little piece here of paper, this silver paper. You have a laser here, and when the nest is vibrating, minimally, obviously, the difference in the return of the signal to the laser is translated in the size of the movement of the nest. Okay, so this is the way that then translates into the ways we can see, and it's quite difficult to use this system because you need to have a very absolute silence, no vibration, specific tables to use that. So we are obliged so far to do that in the lab.
Okay.
And Alessandro, excuse me,
Yeah.
I take the occasion of speaking about methods, then I leave the other question. I was wondering something more about methods, because you told us that you had no time to enter into details for that. But just a few questions. First of all, how did you manage to find the parasites in the middle of nowhere over there? We're looking every piece of stone or something like that. I can not imagine.
Okay.
And also, you mentioned that you mark the workers, any wasp. How did you do that?
Okay.
How do you do that normally?
Yeah. So finding them is not that easy, but as it happens often with animals, if you know where to look, then it's quite easy. Meaning that it's a mix of luck and three things: luck, people, friends telling us I saw one of them, and summer trips. The easiest way to do that is to go in the summer on the top of the mountains where they're mating, and the males they make territories, and they defend these territories, waiting for the females. And so if you go on the top of the mountains, really on the hilltop, on the mountaintop, and you find males there, then you can go back in the overwintering, go back there the next season in the overwintering period and start to remove the rocks. But I have to tell you that sometimes, even in the place where we know that we can collect them, and we're collecting them since 10 years, 20 years, the group in Florence told me that, and they're very expert in that. Sometimes, in some years, we don't find them, and we don't know if we just arrived late or it's too early. They're deeper under the rocks. So it's really, knowledge plus a lot of luck. Coming to the second point, marking, it's quite easy. Colors. We have colors that the colors that you use to do modelism, and you have some that we know they're not toxic. They stay there quite well. And so you make little dots and combination of colors. Actually, you can also use for these species the tag to mark the bees, which they're nicely made and have numbers, and that are really, really better. But they're so concave that sometimes they slip away from the back. So
because you showed a kind of drawing, it seems that you were fixing something like a thread or something. Probably I misunderstood your drawing.
No, probably no. I was drawing on the thorax. Yeah.
All right.
Thorax.
Okay, thank you.
Okay, we have another question from the audience by Lorenzo. Is there any correlation taken into consideration on workers between position in the social network and ABW intensity duration in workers who are getting ready to start the covert rebellion? I like
nice point. I can't say that because we did not record yet the abdominal walking in workers, especially because workers perform rarely this behavior. It's mostly done by the queen and the subordinate foundresses, and it's far less common in workers. I told you it's related to dominance hierarchy. So it would be nice actually to redo the experiment again and see if they're increasing the abdominal walking, and if they do, record the sound, the vibration, and then correlate that position in social network. But no, we have not that data yet.
Okay. And there is another question by Francesco Stefani. It looks like he's very interested in your work. So when the first workers are born, how is the queen able to assess her dominance?
What do you mean by assert her dominance? I mean, the queen, when the workers emerge, is the queen, and they will not challenge the position of the queen. Imagine they emerge, just emerged wasps, and there is a queen that is ready there, powerful. Often the queens start already after emergence to dominate the workers. Then what might happen, however, is that if the queen disappears, one of the workers will replace it. And usually it's the oldest worker on the colony. So the first workers that emerge, they realize by interacting that they are far away in the queue, because there is the dominant individual, maybe subordinate foundresses. And so there is no need to every time put in question, every time establish again dominance hierarchies. And that's also probably why the workers are not openly reacting to the social parasites. Because if you imagine a colony of 20, 30 workers could easily put away the parasite. But they're not doing that, probably because as soon as one emerges, dominated immediately by the other individuals and is the last in the queue. And so it will not try to react, at least openly.
We have another, and maybe the last question from the audience, that is by Gaia di Francesco Antonio. Just curiosity, we have seen that in Polistes dominula, there are more foundresses, but just one becomes dominant. What's the role of the defeated ones in the colony? Do they start behaving like workers, or do they play another role? I would extend the question.
Okay, they act as workers. They when the dominant queen becomes the dominant queen, she stays on the nest, so she's not leaving the nest anymore. So all the tasks are performed by the other workers, the other foundresses, the subordinate foundresses. So basically, they're behaving as workers. But it's not a strategy made just for helping. They're not giving up completely, because it happens quite often that the dominant foundress can die for many reasons, and the second in the hierarchy can take the place. So this has been estimated in the field, and it has been estimated that in many cases, it's better to be the beta, the second foundress in a multiple foundress colonies, than to be the queen of a single foundress colony, because the likelihood of surviving and producing or inheriting the nest is sometimes better than the having a small colony on your own. So they're there to work, but this work gives them the possibility to possibly inherit the colony if that happens. So on evolutionary timescale, these appear to be leveling somehow, and probably that's why they're still there, at least so far.
That's very interesting. There's a one question that I want to take advantage of being the moderator, so I can ask you something. So let me take advantage of my power position. So I guess when we are some of these viewers that are following this seminar are part of our behavioral ecology class in our conservation and evolution program, and we tend to teach them about the comparison, interspecies comparison, to study the evolution of some behaviors, even taking into account the phylogenetic history of the species that we are comparing. So I wonder, do you have any comparable system that is more primitive, to this one, that may support or confute some of your hypotheses? So can you see some of these processes ongoing in other systems, but less evolved, less complex?
Yes. We can see that in within the genus, we have these three species, but they are all like sister species. They're really strictly related, and they have a single origin, so it's difficult to make a comparison there. We can make some comparison, but it's difficult to say who is more advanced and who is less advanced, etc. But to see something more primitive, we could go to look for facultative social parasites. So those species within the same genus then can do the nest on their own, but in some cases, they decide to usurp another colony of another species, or also facultative intraspecific social parasites, that instead of founding their own colony, they usurp the colony of another of the same species. So we have different stages that probably, according to some, are the way through which these social parasites evolved, from being facultative parasite within their species, then going to another species, and then being obligate. So we can go there and try to test that. The problem is that it's easy to know that a social parasite, obligate social parasite, it's a social parasite because it's a species. When you go to a facultative social parasite, it's far more difficult to be sure that you take an individual that's going to be a facultative social parasite. So you can have a test and wait for it to show that it wants to be a facultative social parasite, but it means having thousands of wasps in the lab, so it's not so easy practically. but it's feasible in principle. And we tried to do something of that, and came with mixed results. So I'm not introducing the talk because it's few samples for the reason I was just telling, but yeah, it's the avenue that the next line of research should be that one, if feasible.
Could you do selection of certain features to test your hypothesis, meaning individual?
Evolutionary selection, you mean, like natural selection, selecting them?
Yeah, you as we have seen in other situations, when you want to test whether a certain trait is very efficient in manipulating or facilitating some of the parasitic behaviors, and I assume you have variability in the parasites. You may select those that show extreme values of those traits. Have you ever tried, or have you seen any?
I dreamt about that, but I never tried, because there are two practical problems. One is that it's not easy. It's easy to rear them in the lab, but not to have them mating in the lab.
Okay.
So people tried several times, but it's not easy even to see reproductive behavior. So imagine to have a proper mating. But I mean, it's a practical challenge that can be overcome. Another challenge is that for the social parasite, they do these altitudinal migrations. So they go to the top of the mountain and coming back. And I guess that it's quite important in their life cycle. So we might do something fake, but so far we never tried, because on a short time it's going to require a lot of effort. But that would be a dream, to be able to select them and do have different lines of big head parasites, small head parasites, those who are good in mimicking, those who are not good, etc. Yeah, that's why at some point I thought I could change the field, because it was not possible to do that. So I was thinking maybe I have to change the model organism.
Anyhow, it's great, and I don't know if Paolo has a very last question, or we are lucky because now, when this seminar is finished, myself, and the class of behavioral ecology, we will move into another platform, and we will keep questioning like hell Alessandro, and have fun in discussing with them. But for the sake of this seminar, I wonder if you, Paolo, want to say a few words for closure and thank our guest.
Well, I would have a final question, because it was one of the main one, but I put on the back. I was surprised by your finding that the parasite queen provides a special signal, this vibrational signal, which with many very different features, like what it was, more longer, and more intense. And it was very much longer and very much more intense, which, I mean, isn't it, could it be a bit, so to speak, risky for the parasite to send a signal that is so far away from the natural range of signal? I mean, general parameters, features like duration or intensity are used to differentiate communicative signals between species. So it might be that if you give a vibration so long, it might be not recognized as a species-specific one. I mean, it's quite, because it was about double as long, or something like that.
It's surprising, and it was exciting when we recorded it. Actually, it was known from several years behaviorally, but it was really nice to record it and to demonstrate that. I agree with you, but we have also to remember, about the superstimuli and the fact that, like, for example, in cuckoo birds, you have this like red colored throats, or the aggressiveness elicited by red stuff. With a red pencil in front of an aquarium. So it's surprising, but I really hope that it's a sort of super stimulus for showing, because actually, if you do that, as you told at the beginning, it's risky. It's also energy demanding to do that. Sure. I mean, it's costly. So it might have a reason. And probably if this intensity is related to dominance, by showing that you are able to beat the nest strongly, you're probably showing to the larvae and to the other individuals on the nest that you are there, there is a proper dominant individual, and nobody needs to be rebelling. Them they probably do as well.
So,
but
so you're right. You're probably found. You're correct. No, I was thinking to this. My following question would be that one.
Yeah.
It seems like so these parasites are acting like nicotine bergen, providing big eggs to the oyster catcher or something like that. All right, thanks.
Yeah, I hope so. I mean, it seems so. We have to do the playback. We tried it once. We did not manage to finish the experiment for practical reasons. But we're trying to do that to see if they're really if there is a response. Yeah, I hope so.
Okay, then I would like really to thank Alessandro for his seminar. And you have 5 minutes of physiological break, so if you need to. And we wait for you in the other platform. I sent you an invitation.
Okay, perfect.
Let me know in case you don't receive it, and I will send it again, okay?
Great.
So thanks, everyone. There is another question by one of our students, but he will provide these questions to you in the other platforms, so we can make a closure of this seminar. So thanks again to you, and I hope we will have as many people and even more attending our seminars on March the 17th at 2 o'clock, when Professor Claudia Carrere of the University of the Study of the Tuscia will talk about animal personalities, the power of the individual. So thanks again to everyone, and see you soon.
Thank you. Bye.
Bye.